topic is updates in veterinary clinical parasitology. Dr. Sangren asked me to talk about advancement in veterinary clinical parasitology. I was a little bit confused about the word advancement and update, and I thought I should stick with updates because it is easy to do that rather than doing an advancement, which I know there are lots and lots of advancement happen, but I work in a diagnostic lab. It's kind of an academic commercial diagnostic lab where the reality is like we don't use a lot of those advancements. So I thought
Mani, sorry to interrupt. Mani, can you increase your voice a little bit? Because it looks very feeble for us.
Can you hear me now?
Yeah, it's better now. Yes.
Okay. Let me straight away start by telling where I work. So I'm at Cornell University, as Dr. Sangren mentioned, at the College of Veterinary Medicine, and we host the New York State Veterinary Diagnostic Lab. Let me use my pointer to show. So that's the New York State Veterinary Diagnostic Lab, which is also called as the Animal Health Diagnostic Center. We have around 13 sections inside, and one of that is Parasthology, and I'm the director for the Parasthology section. So we handle around 225,000 submissions every year, and in Parasthology, we handle around 12,000 cases every year. So before I jump to my talk about updates, I just wanted to use these two slides to tell you about a small history about how veterinary parasitology evolved in North America. So the person here on photo is Dr. Salmon. Like, you know, there's bacteria, like microbiologists are aware of Salmonella. So this is the person after which like the bacteria was named. So he was the first veterinarian. He graduated from Cornell. He's the first veterinarian in North America, and he was made the chief of Bureau of Animal Industry. And during that time, 1884, there was one important disease here that occurred at Texas. So Texas is a large state here in North America. So in that state, like lots and lots of cattle were dying, and they called that as Texas cattle fever. So at the time, many people were trying to identify what is causing the Texas cattle fever. And under the leadership of Dr. Salmon, there was a team formed, and many of the scientists who worked in that team were graduates from Cornell. And I have listed here a few: Dr. Smith, Dr. Kilburn, and Dr. Curtis. These three people were key in that team, which worked and solved the mystery behind Texas cattle fever. So what they found was the ticks called the Boophilus annulatus. Like now they renamed that as Rhipicephalus annulatus. So this tick was responsible for transmitting the disease caused by a protozoan parasite called Babesia bigemina. So they were the first to identify that a vector, in this case, a tick, is an intermediate host for Babesia. It was not known before the time that vectors like arthropods can transmit disease. So this trio were responsible for identifying that ticks can act as a vector or as an intermediate host for transmitting disease. But in this case, like it is an animal disease. So it happened way before in 1890, and they published their report as a book called Texas Fever. So that book is still available. Like I have it in my desk now. So they were the first. But in 1898, Dr. Ross from India, like he identified mosquitoes as the vector for transmission of malarial parasite, and he was awarded the Nobel Prize in 1902. But if you look at the history, it was much before Ronald Ross' discovery. The trio from Cornell, like they identified ticks as a vector for transmission of Babesia bigemina. The concept was just discovered by people here in North America. And among them, Dr. Curtis, like he was responsible for identifying the life cycle of Babesia bigemina involving ticks. So he was aptly called the father of veterinary parasitology in North America. So once they discovered that ticks were responsible, Then they started this cattle fever tick eradication program where they formed this dipping vat where they allowed all the infected cattle to pass through. So periodically, like every 14 days, animals were made to pass through this dipping vat, and that kind of kill all the ticks that the animals were holding, and that was the main idea behind controlling this cattle Texas cattle fever. So there were a lot of publication that arise on this aspect, and there's one book that was published by USDA, and they named it as a canal to prosperity. And this is this U.S. Dipping station where all animals were allowed to pass through, and so they shed the ticks, and that's how they control. So this success today, if you do the valuation, it saves around $3 billion every year in savings for cattle industry in North America. So with this brief history, let me jump to the actual topic updates. So before I do the update, I just also want to introduce another person from history, which is Dr. Hall. And he is also considered a father of American veterinary parasitology, along with Dr. Cooper Curtis. So he worked essentially on canine hookworm, and he worked on finding out a lot of drugs that can act or that can be used to treat and control canine hookworm disease, and a lot of publication on this. And he was also mentoring lots of students in veterinary parasitologists he trained, and those all became a big leader in the field in coming years. So the reason why I'm showing him here is to tell that during his time he used microscopes, which were as you see here, the old microscopes. So looking through that microscope, he identified these canine hookworm eggs, and years passed by, like almost a century now, like we are away, and we still use microscopes to see the same parasite stages in any of the samples that we examine. So if you ask about advancement, I would say technology advanced, but whatever we are looking for parasites, that's all the same. So here you could see a high-end microscope with cameras attached. We use still these microscopes to identify the same parasite that he was looking 100 years ago. So this is from my lab, and I just wanted to use a canine fecal diagnosis as an example to show how advanced we are, what advancements we have made so far. So in this, like you would see, there's a list of common canine parasites diagnosed in fecal submission. So these are the common parasites that from my lab we generally identify on regular basis. In this, I have highlighted some in red. So those are the parasites for which we have advanced diagnostic methodologies. For example, LISA, or lateral flow assay. So the lateral flow assay is developed by a private company called IDEX. So IDEX use lateral flow assay for identifying some of the parasites like Toxocara canis, Trichuris, and Chylostoma caninum. So those are the parasites that are more frequently encountered, and we have lateral flow assays for identifying those. And parasites like Giardia and Cryptosporidium. So we do have ELISA, and for example, for Cryptosporidium, we do have ELISA to identify parasites to the genus level, but not to species. And for Neospora caninum, a lot of folks use indirect fluorescent assay, which is again a common test. But apart from these highlighted parasites, we don't have any other advancement for other common parasites that we still encounter in the field. For example, Cystisospora canis or Ohiensis-like parasites, we do see them a lot from shelter dogs, but there are no advancement made. Like there are current efforts directed towards developing ELISA or lateral flow assays for some of the parasites, but it is a long way to go. So what are we doing in the lab currently? We still use the time-trusted old classical parasitology methods like fecal flotation test. And you could see here, these are the trays of fecal flotation slides that we do in our lab on regular basis. So on one day, we had around 150 to 200 samples are needed, and we all process them by classical techniques like Wisconsin double centrifugation flotation, which I guess like you guys in third year would have some experience doing yourself in your lab. So, but that's the technique that we use in America for diagnosing parasites in not just dogs, but in all other hosts, like a wide variety of hosts. We still use the fecal flotation method. So if you ask about advancement, this is where we stand. So on parasite diagnosis, we talked about fecal submission in the previous slide, but on the parasite diagnosis, like doing a gross examination, so we still use microscopy. The only thing like that differ is like we process the sample. For example, I have here a lice that was collected from a bald eagle. So we process them, but we need to look under microscope to identify what it is. And there are some nematodes, like we process them in lactophenol and look at the features, morphological features, to identify what they are. And a lot of times, like pathologists, like they do the section, and then once in the section, if they suspect for a parasite, they try to identify what it is, or they seek the help of parasitologists like us to make the diagnosis, and we help them on regular basis. So what I'm trying to tell here is like even the parasite diagnosis, like gross morphological identification, so we still trust our classical techniques like microscopy to do the diagnosis. Like it is, I mean, in terms of advancement, not much has happened, but still the classical methodology is the trusted technique for getting your diagnosis. But if you wonder, like, when is molecular diagnosis employed in a commercial or even in an academic diagnostic lab like us? So we prefer to do PCR only when there is a doubt. For example, these are the two parasites collected from a fish intestine, and one is Diphyllobothrium, another one is an Acanthocephalum. If you look closely, um, in Acanthocephala, like you have a proboscis that extends at anterior end, and sometimes this proboscis is not protruded outside, and it is like retained in. And if you look at this specimen and compare with a free pleurosarcoid, like what I show in here, so it is really hard to differentiate these two. So what to do now? Like classical methodology is not helping you to identify what they are. In that case, we would try to employ PCR, and then once PCR is done, you have the results on the sequence results on hand. You blast it, and it matches with what is there in the sequence database, and you know the results. So in a diagnostic lab, a commercial lab, like we try to use PCR to complement what we do on the classical aspect. So with that, like I just wanted to give few examples. This is some sort of research that happened in our lab when I was working at Calgary. So I just wanted to show you like how we blend the classical methodology for the parasite diagnosis with that of molecular, and try to come up with the diagnosis. So this is one case of surveillance that we did for one of the important tapeworm called Echinococcus multilocularis, which is common in coyotes. Coyotes are kind of dogs, like wild dogs, which is a definitive host similar to red fox in Europe that can host the parasite Echinococcus multilocularis in their gut. And they shed the eggs in the feces, and the eggs are eaten by the intermediate host, in this case rodents. And the multilocular hydatid cyst can occur in the liver, and if the definitive host consumed, the life cycle goes on. Humans are accidental host, so it is a zoonotic disease, and once they ingest the eggs that are in the environment accidentally, then they suffer from what is called as alveolar hydatidosis. So we did the surveillance in two cities in North America, which is Calgary and Edmonton, situated in the province Alberta in Canada. So what we were aiming at is to see if the coyotes that are present in urban settings, like in the core urban area, host these parasites or not. So when this project was started by an ecologist at University of Calgary, he collected dead or the roadkill coyotes from the city, and then so we helped with the necropsy, collecting the guts or the intestinal tract from these coyotes and started examining for Echinococcus multilocularis. So here you see these are the tiny Echinococcus multilocularis that we collected, and I put it in a vial and onto the scale here, and they are so tiny, and you need to use a microscope, a stereoscope, to identify they are what they are. And when you look under the microscope, you could clearly see a tapeworm. This is the whole tapeworm with the head end here and the gravid segment on the other side. So if you do the measurement and if you look at some of the morphological feature, so by just looking at them grossly, it is possible for us to identify them as Echinococcus multilocularis. And if I wanted to publish this, and if I just use the classical way of identification and try to publish them to journals like Emerging Infectious Diseases, they won't accept it. Like that's how the trend is now. So they would ask for PCR or molecular confirmation. So at that time, like I used the more traditional conventional PCR to rule out Echinococcus multilocularis from another common Echinococcus, which is granulosus, which is also coyotes can get infected with. And then the third one is tinea. So tinea is a broader tapeworm that like you could encounter in coyotes. So all these three tapeworm shed eggs in the feces, which looks the same, the tinea egg that I showed in the previous slide. But unless like you collect the parasite and examine yourself, it is really hard to differentiate these three. But in my case, like I know that I have collected a parasite which is smaller than Echinococcus granulosus or tinea, but still publication, if you want to make, you need to do a PCR. So in this PCR, like I just don't want to explain in detail what it is, but what I'm trying to show here is our sample like amplified a segment, a region in the ND5 segment, which matched with positive control, and we know this sample is Echinococcus multilocularis. So we confirmed this, and we did a further PCR to identify the strain, which is a North American strain of multilocularis, and then we try to publish this in Emerging Infectious Diseases. So I to just touch on the results that we got, like around 23 of 91 coyotes that we examined had Echinococcus multilocularis, which is 25%, which is a good, like a surprising number for a coyotes that lives in the cities to carry this zoonotic disease. So we published this, and then it was well received. That was the first report about Echinococcus multilocularis in Alberta. Another case that I just wanted to highlight here, again using blending both the classical and the molecular parasitology, is about a parasite that lives in the eyes of dogs, which is Onchocerca dupi. So you could see here on the subconjunctiva, like you see a nodule, and this nodule, like if you tease apart, you could collect the worms from there. So these worms, they I mean, the worm is usually, this parasite is usually common in Europe, and it slowly emerged in US, in the United States. And then a few years ago, like when we were at Canada, like we saw a few cases of this parasite occurring in dogs, which has travel history to the US. So the traditional way of identifying them is to make a skin biopsy, skin snips from here, and then soak it in saline. And these parasites, they shed the microfilaria, which are present in the skin around the eyes where the infection occurs. And soaking this skin snips in a saline, you would see the microfilaria or the larvae just moving inside the saline, and then you could examine them and do all the morphology to identify what they are, to confirm what it is. On the other hand, you could also, like if this nodule is not visible, that's when you test for microfilaria. If this nodule is visible, it's easy. Like you just take the piece of the parasites from here, and then you can examine by classical methodology, or you could do a PCR. Let me explain why classical is important, because it is time saving compared to molecular. So on this slide, I'm just showing a section of the mid body of the worm that we retrieved from the eye of a dog. And then if you look closely on the cuticle, so you look here, so this is the outer cuticle, and then you see here a ridge sort of thing. There are two ridges on the outer cuticle, and then on the inner layer, like you see, there is two projection. So this is the pattern, like in between these two long ridges, like you would see two um, uh, like arrow-like pointing projections. So if you look at like here, there is another ridge and then two pointy projections. So this is the pattern very specific for Onchocerca lupi. So if you look under a microscope, by collecting this parasite, keep it on a slide, add some lactophenol for clearing, and then look under a microscope. Within maybe less than 5 minutes, like you can identify what this parasite is. But if you're not aware of how to do the classical methodology, then you would just do a PCR, but PCR is time consuming. At least it takes 2 days for you to get the PCR and the sequence result to compare and find what this parasite is. So in terms of advancement, yes, like PCR can be used, but if you are a trained parasitologist, you could get this results in 5 minutes. So, but what PCR can do? So once you do PCR, like you get the sequence, and then you can do this phylogenetic analysis. And in this case, like we just wanted to identify how the sample that we collected from Canada matched with all samples that we know from Europe as well as from US. So in this case, like we clearly show that the Canadian samples matched with what is known from US, and this clearly established that the worm or the parasite that we encountered in Canada is very similar to the one that is occurring in the US. So for this, like yes, advancement really helps, but in terms of diagnosis, clinical diagnosis, like we still trust the classical parasitology. Another recent example that I wanted to highlight here is Thalacia calipeda. This is another cool worm of dogs. Like here you could see the worms that are infecting the dogs. This is different from Onchocerca, where Onchocerca forms the nodule inside which the parasite lives, but these worms are free living on the eyes. It is again of zoonotic concern. So fruit flies act as an intermediate host and transmit the disease to human eye, and the life cycle goes on. So if you look at the distribution of this Thalacia calipeda, this is also called as a oriental eyeworm, very common in places like India and in East Asia. And 30 years ago, this was introduced in Europe, and it spread really quick in almost all of Europe. And in North America, there was no report of Thalacia calipeda. And the equivalent of Thalacia calipeda is Thalacia californiensis. As the name sounds, californiensis parasite is very common in the California state of US, but not common in all states. But until we reported, like there was no report of Thalacia calipeda in the US. So how we made that diagnosis. So here I've shown the anterior portion of Thalacia californiensis, which we frequently encounter in North America. And then you look at here, this is the anterior portion of Thalacia calipeda, which we recently encountered. And classically, you can differentiate these two. So these two parasite images are taken at the same magnification. And if you look at the annulations here, so the ring sort of thing that you can easily see on this parasite on the anterior side. So if you count that ring, like you may count around 110 or less of those rings in 1 millimeter length of the parasite. On the other hand, if you look at the annulation, they are very tight and close enough here. So if you do the same, like in 1 millimeter of this parasite, you would count more than 200 annulations per millimeter. So that's a simple key that you could use to differentiate Thalacia calipeda from californiensis. Again, if you want to publish, like journals won't accept just based on classical morphology. They would ask for further PCR details, and we did PCR on this specimen, and you could see here that's the that's where the New York or the US specimen matching with Thalacia calipeda that were collected from places in Europe. So it clearly shows that Thalacia calipeda that we encountered here in the US came from an European or has its origin in Europe. So we recently published this, or the article is in Press in Emerging Infectious Diseases. Moving on, another example or another update about a parasite that for which like a clinical diagnosis made the difference. So this is a tick that we collected from, not just we, like our collaborators from another university collected, and then they once they try to identify based on the keys available in North America. They've sensed that this is a different parasite, a different tick that we don't commonly see here in North America. So this parasite, this tick, is called as Hemophysalis longicornis, or the Asian long-horned tick. If you guys are aware of some of the anatomy of tick, so this is the basis capitula, the whole thing, and then you see the palps that are projecting from the basis capituli, and then on the third palp, like you could see a horn-like projection pointing backwards. So this is that projection. So this is common in Asian long-arm tick, and then some of our collaborators they found this, and then they suspected this as Asian long-arm tick, which was not reported here in North America. Then they sent the sample to the reference lab, and they did a PCR and they confirmed it. But looking at the morphology was the first step in identifying this exotic tick here in North America. So once this identification was made, then in the last 3 years, like the surveillance efforts were increased, and you could see here in the northeastern part of US, like a lot of counties. Those dots here are not, those highlighted here are the counties, and you could see many counties, like more than 96 counties as of now, report the presence of this particular tick here in North America. So this tick transmits Theileria orientalis, which is a protozoan parasite, and again, it is an economically important disease in cattle, and this tick can transmit this particular disease for cattle in North America. So the surveillance efforts are still on. Again, so we are still working on to find out the actual spread. And at this point of time, it seems like it is concentrated in the northeastern state, except for a small one county in Kansas. It has not spread to all of the part. Like, we are trying to contain this, but I don't sincerely feel like it is possible for us to control this tick at this point of time. So that's another update. And to move on to the last one that I want to highlight here is how we use classical technique to identify some of the tapeworms that we encounter in the field. So if everyone is aware of how a tapeworm would look like, it has a scolex and then all the strobila and proglottids behind. And here is where the scolex is. And if you like look under a stereoscope, this is how the scolex would appear, with some of the parasite holding the hooks at the anterior end. So if I'm doing the classical identification, I would prefer to dissect the hooks on the anterior end, like how I have done here, and then study them, or do the morphometry of the hooks, like that is like studying the morphology as well as the length and the width on all those features, and which like you could use to compare with the keys that you have in hand to identify what parasites they are. So this one, I did it when I was trying to learn this technique. So you could see I didn't do a perfect job. I didn't do the cut exactly behind the line where the hooks are. So it was not that clean, but still it helped me to do the morphometry. So here is a typical hook, how it appears. So it has what is called as blade on this side and a handle on the other side, and this projection at the bottom is called guard. So this is typical of a tinid hook, and if you do the measurement, and you could and based on the shape and the measurement, you could match this as a Tinea pisciformis. So almost all the tapeworm hook look like this. Like in this case, the blade is shorter than the handle. So this is the typical way, except for one species, Tinea crassiceps, in which, like if you look at the handle here, the handle is shorter and the blade is much longer. So this is the one exception where you see a much longer blade compared to the handle. So what we encountered here in New York, once like there was a submission, a muskrat which was killed by, it's a roadkill, and it was submitted for necropsy. On the necropsy table, when they opened up, they found these white dots all across in the abdomen, and the pathologist they did a histology, and then they noticed that it is a metacestode, which is the larval stage of a tapeworm, and then they noticed some hooks in here, so you could see the hooks and the suckers on both sides, and they know this is a metacystode, but they wanted an ID done, and that's when this specimen came to our lab. And the first thing I did is take one of those white dots here, like you see lots and lots of white structures. So I took this white structure, pressed under two slides, and then I looked for the hooks, and you could see here there is a large handle compared to the short, a large blade compared to a short handle. So I know based on morphology this is Taenia crassiceps. But again, we did a PCR to confirm this as Taenia crassiceps, and we reported this as a first occurrence of Taenia crassiceps in a muskrat in New York State. So these are some of the examples that to show that in terms of advancement, we in a commercial diagnostic lab still use traditional classical methods to make diagnosis, but all the advancement that you hear in the field, it's still at its infancy in a commercial lab. So what I'm trying to say here is, in reality, like not much of the advancements made in parasitology are applicable for doing routine diagnosis in a commercial lab. Despite that, I just want to highlight on two of the recent advancements that we are trying to develop in our lab. One is tick-borne, doing a nanoscale Pcr for tick-borne disease identification on a high throughput scale. This is one thing that we are involved in. Another one is identifying the gastrointestinal Nemo Biome using Manigandan approach. So let me just explain what the tick-borne disease high-throughput surveillance with nanoscale Pcr is. So this program, this evaluation program, occurs at the Animal Health Diagnostics Center where I work. And what we do is our clients submit tick. Ticks are a really scary thing here. So they one of the disease that one of the main tick that carries here is Lyme disease. I don't know if you guys are aware of. So it's a very pathogenic disease that a lot of people suffer from. And for that reason, if anyone is bitten by a tick, they collect the tick and send it to us. Or any dogs, like or not just dogs, any host have a tick on them, then it comes to us for tick identification and further evaluation for the pathogens that they carry. So the first step when a tick is submitted is it comes to my lab, to Parasthology lab, and I'm responsible for identifying the tick to species. So we have a key to identify the 5 common, usually was 4 common species, but with the addition of Haemophilus longicornis, now it is 5 species. And what we do, or what I do, I just look at the morphology of the tick to identify what tick species they are. So it's very easy to do the tick identification based on morphology. So here, this is the ventral aspect of a tick, and this is the anal opening. And you see here there is a groove that goes anterior to it. And on this is another tick where there is anal opening and the groove appears posterior. So based on where the groove goes, like you could able to broadly classify 2 groups. One is ixodis, another one is non-ixodis. So anything that has groove anterior, we call this ixodis. So ixodis capillaris or ixodis pacificus, all those ticks carry this groove anterior. Dermacenter, amblyoma, or rhipicephalus, those are non-ixodis tick, has this groove posterior. Very simple, easy way to differentiate species. And then we use other features, like for example, this is basis capituli, and you see the projection of basis capituli, which is different than if you look at the basis capituli, the projection is lacking. So if the projection is present, this is rhipicephalus. If the projection is absent, this is dermacenter. Again, dermacenter, you look at the palps are shorter than the palps of amblyoma, which is longer. So these are simple features which you could use to group the different important genus of parasites. And here on the top, like I have here, there is a palp, and the second part segment of the palp is projecting laterally, which is typical of Hemophysalis, which is absent here in Derma Center. So there are keys available. Like if you look at George's book, like we in the recent edition, like I have provided the key for how to identify ticks, the common genera of tick. And if you use that key, it's a matter of five minutes or less than 5 minutes to identify what tick species you have. So once my lab identifies the tick, then the tick is then sent to the molecular section, which is another section working within Animal Health Diagnostic Center, and they are responsible for identifying what the tick pathogens are. So before I move on, like just wanted to highlight on what are the common species. As I said, like Ixodes scapularis is the majority of the tick that is submitted to our lab, followed by Dermacentor variabilis, which is the American dog tick, Amblyomma americanum, the Lone Star tick, Rufficephalus sanguinus, which is the brown dog tick, and Ixodes scapularis, again, that's the deer tick. And recently we had Hemophysalis longicornis added to this list. So these are the common tick species that we identify in our lab. So the diseases that this tick transmit, the most important one is Lyme disease caused by Borrelia burgdorferi, which is transmitted by Ixodes scapularis. So highlighted here in red are those diseases that Ixodes scapularis can transmit. And Amblyomma, they have their own vector potential for transmitting Ehrlichia and other similar parasites. And Powassan virus, again, Ixodes scapularis can transmit that. And the reason one, longicornis, like they transmit Theileria orientalis and severe fever, thrombocytopenia syndrome virus. So these are the virus and protozoan transmitted by Hemophysalis. So based on the tick that I identify in the lab, I send the tick specimen to molecular section, and molecular section based on my tick identification. Then they have their own panel. Like how you see here. So for if it is Ixodes, they have their own panel of PCR for various disease like anaplasoph, Lyme disease, and then the Bisha microti. So all those based on what tick species that I identify, like they use the panel and do the PCR for the specific pathogens that this tick species can transmit. So this is how the tick-borne disease test panel or just test evaluation occurs in our lab. But we had a discussion and we thought, like, can we do away with this panel because it's time consuming? Like you just need to identify the tick, and then you just need to identify which PCR to do based on the tick species that you have. So what is the advancement here that could help to do away with the panel? And this is what is called the nanoscale PCR. So to admit, like I am not a molecular biologist. So this work was done in collaboration with Dr. Laura Goodman. She is the molecular section director, and she is well versed with all these techniques. And my part is tick identification and trying to see what pathogens they are testing for. I do a sincere attempt to explain this to you, and if there is any question, like I would prefer to redirect that question to Dr. Laura Goodman, who is our molecular section director. So what is happening here is like this nanoscale PCR, it occurs on this large chip, and it has a lot of these cool structures. So what they are trying to do here is they pump 15 to 30 microliter total reaction volume into each of these small holes here. So these pores here, like that's where they pump this 15 to 30 microliter reaction volume of PCR material in there, and then do the sequencing. So what they let me move on to the next slide to just show you how exactly it is done. So once the ticks are sent to the molecular section, like they do use these beads to extract DNA from each tick, and then once they collect the DNA, these DNA are loaded onto this chip, in which like each square here is for one sample. So you have around 48 such sample holder here. So 48 samples can be run in one single PCR cycle, and then they throw it in this high throughput sequencer, and it does all the work. So what we were trying to design is a panel, a nanoscale panel with all the 18 pathogens which ticks can transmit. So in this, like 18 pathogens, and I listed here what those pathogens are. So if you look at this slide here, there is one square here. In this like square, like there are 18 holes, and each of those pores, you could load them with probes specific for each of the pathogens that I list here. So, and they are run in triplicate. So you have here 1 to 18, and that's one thing, and this is the duplicate, and that's the triplicate. So once you have things run in triplicate, then you start extracting DNA from each of the tick. And as I said, like you could run 48 sample. There are 48 such squares here. So you put the DNA of 48 such sample into this chip, and then send it to the sequencing, and it gives the results. So we compared our nanoscale PCR results with the ones that are traditionally run, and here is the data. So this is done not by our lab, but the New York State, and they tested around 7,000 ticks, and 46% of them carried pathogen, and that's the diagram that shows the extents of different pathogen distributed in these ticks. And our nanoscale PCR, which we did with 904 ticks that was submitted to our lab, and we still get a comparable results of what is identified by the New York State, like Department of Health. So we see a comparable result. So the nanoliter PCR yields a comparable results. So this is one thing that we are still working on, and our idea is to use this for high throughput when there is a large tick submission, not just one or two ticks, like thousands and thousands of tick, like research labs or any big projects, like we wanted to employ this nanoscale PCR to get a result in a quicker time frame. So that's one thing. And the last thing which is happening in my lab, and I'm involved with this analysis, which is using a Manigandan nanoscale PCR to identify Nemo Biome. So just to give you a brief background on what Nemo Biome means. So you do a fecal examination for example, this is sheep, and if you do a fecal examination, and you would find these typical eggs, we call them as strongyle eggs. These strongyle eggs could be from any of the parasites that I listed here. So basically, we call them as strongyle because it's really hard to differentiate morphologically the eggs of Ostratagia from Himancus or from Cuperia. They all look the same, and that's how the eggs would appear in a fecal float. So it is really hard to identify what these species are. So if you ask the question why it is important for us to identify the species, it's the reason is like there are some parasites like Himancus, which are blood-sucking parasite, and they are of real concern to sheep raising. Compared to Cuperia, they are non-pathogenic. So in order to identify if pathogenic parasite is present there in an animal or not, like we may need to identify that two species. And what is the classical way to identify that is called as nematolobal culture. So here are the steps. So you just need to basically mix the fecal sample that you collect from a sheep with vermiculite, and then leave them in culture for 14 days. And at the end of the 14th day, what we need to do is like fill this cup to the brim with lukewarm water, and then put a petri dish on top, flip it over, and then pour lukewarm water on the outside, and then leave it for another 2-3 hours. All the larvae, when I say nematolobal culture, the eggs would hatch to stage 1 larvae, which molds to stage 2, and then the final mold is stage 3. So those third stage larvae are present in the sample, and they would migrate to the water which is on the outer like petri dish. So if you keep this setup for 2-3 hours, all the mobile or motile larvae would come to the water that is present. So you could able to collect the water containing the larvae, and then use the key here. So I have here the key for the third stage larvae. So you could do all the measurements and other things to identify what they are. In this case, this one is Himancus contortus L3 stage 3 larvae. Frankly, this requires some good expertise. Like you have to get yourself trained to identify what the stage 3 larvae are, whether it is Himancus or how you could differentiate other things like strongylitis or Copelia. So these are based on the tail length and on all other features. So I'm not going to go in detail, but it is possible for anyone to do an larval culture and identify all the parasites based on classical methodology. But to be frank, like it is not possible for you to identify to species, but at least to genus level, it is possible to do an larval culture and identify them. But in reality, mixed strongyl infection is the rule. So any sample that you collect and do a larval culture, you just don't find single species. You find a mixed species. Like you may have Himancus along with Cuperia or Ostratagia. So that is a common reality. And is there a way that you could do an accurate identification of all the strongyls present? And is there a way to quantify them? And the answer is yes. But how we could do that, I'm going to explain in the subsequent slides. For that, like we just need to identify the region that we need to target for some of the nematodes here. So the strongyls that we talked about in the previous slide, they all fall under clade 5. So they are clade 5, and morphologically, the clade 5 nematode, these are adult strongyl, has synlophs. These are the longitudinal ridges. Like if you could notice here, there are longitudinal ridges. So if it is present, we call that a synloph, and all those worms that are, or the helminths that has synloph, we classify them as a member of clade 5. And here is what the clade 5 is. And two examples here is Trichostrongylus brunostomum and Himancus. All those are present under clade 5. And all these clade 5 nematode, we have information about the ribosomal RNA gene. So that's good thing. So we know the sequence information of ribosomal RNA gene for this. And now with this background, like let's see if it is going to help us with identifying the Nemo biome. So when I say the Nemo biome, it is all the strongyl worms present in the gut of a sheep. We call them as a sheep Nemo biome. So can we identify that? For that, let me take you to the next slide to little bit describe about what the ribosomal RNA are. So again, so this one, it is the ribosome unit, like it has both small and large unit, and it is made up of RNA. I have here a whole list of RNA here, and then the protein. So these are structural RNA, and the proteins, they together make the large and the small subunit. So on the large subunit, you have a 3 structural RNA, which is 5.8S, 5S, and 28S, which makes the 60S large subunit. And on the small side, like you have 18S, and both together forms the ribosome. So the ribosomes are protein containing structural RNA. So these structural RNA, the code for this structural RNA is present in the DNA, and there is a pattern how this is present. So you see here the coding information for 18S is followed by 5.8S, followed by 28S. And in between this coding region, there are some non-coding region, or the internal transcribe spaces. We call that ITS. ITS1 is between 18S and 5.S, and ITS2 is between 28S and 5.S. So these are conserved region. The 18S, 5.8S, and 28S are really conserved region across all nematode species, but the ITS1 and ITS2 are really variable. And this ITS2 especially is variable in the sense like Himancus has its own ITS2 sequence, and Cuperia has its own ITS2 sequence. So if we amplify this ITS2 and then sequence it from any DNA that we extract from a worm, it is possible for us to identify the worm based on the sequence information for ITS2. So to illustrate what I mean as conserved or non-conserved, so I use Trichostrongylis and Brunostoma species, and so this is the conserved region, and then the variable region, and followed by another conserved region. So you design primer based on the conserved region. So highlighted here is the ITS2 forward primer, and then on the other end of the variable region, so another conserved region. So this is the second primer or the reverse primer. So now I have two primers, ITS1 and ITS2, and if I use these two primer and amplify, and based on the variable region that is present here, if you sequence and then read that sequence, like, it is possible for you to identify whether it is Trichostrongylis, Bunostomum, or in that case, any strongyle worm that you have in hand. So if you want to identify a single, like identify the identity of a single worm, this is how you do. So you do the DNA extraction from the worm and run the PCR using ITS2 primer and send it for Sanger sequencing. And once the sequence results come, you blast it and you know what that species is. And if you look at the sequence information that comes from Sanger, this is how it comes, and you read the sequence and blast it, and whatever it matches on the top, and that's your species of interest. This is just for doing a single clade 5 nematode identification. But we have a situation where we wanted to identify the Nemo Biome. We are not interested in identifying single species because it is tough to collect an egg and then do a PCR and identify that way. But we have a whole list of GI nematodes. We call that as Nemo Biome, and we want to identify all the species that is present. And on top of that, you in a herd situation, so each animal in a herd have their own Nemo biome. So for example, I have here animal 1 which may have 5 strongyle species, animal 2 has 7, or animal 3 has 4. These are hypothetical situations I created to explain how this is done. So I want to identify all the strongyle species present in each animal and in all animal in a herd, and using the PCR for ITS2. And one method that we could use is what is called the deep Amplicon sequencing. So with deep Amplicon sequencing, it is possible for us to get the information about strongyles present in each animal, and again in all animal in that group. And let me explain to you how this is done. So first of all, we need to identify the animals that we are using for this test. We use barcodes for each animal. So as I explained here, so this is the ITS2 region, which is variable for each parasite, followed by the conserved region. And ITS2 primer, we call that as NC1. This is the forward NC1 primer, and this is the reverse NC1 primer. So what you could do is add barcodes specific for each of the animals that you collect the DNA or collect the samples from. So what I do is take the fecal sample from animal 1 and separately animal 2 and 3. And then I could either extract the DNA by a flotation method, or I could extract the stage 3 larvae, or stage 1 larvae, or stage 2 larvae by the nematolarval culture. So once I have the necessary material in hand, I extract the DNA from each of these worms separately, and then I do an NC1, use the NC1, NC2 primer and run the PCR separately. And once I have run the PCR and collected the PCR product, then the next step is I would add the barcodes separately. On the other hand, you could attach the barcodes to the primer itself. So you add the barcode for animal 1 to the primer itself. And when you're doing the PCR, you just use the barcoded primer for sample 1, and second barcode for animal 2, third barcode for animal 3. So you have all the barcoded primers, and you do the PCR. So these are barcodes specific for animal 1, the green ones, and these purple one are specific for animal 2. So like that, you use the barcodes and then amplify, and now you could group all the barcoded primers together. And the next step is you need to sequence all this. And for sequencing, there are two approaches. Like either you use Illumina sequencing or Manigandan sequencing. For that, you need to add the adapter. So the adapters are it would help the barcoded primers to be sequenced with Illumina or with Manigan sequencer. So either you can add the adapter at the after you add the barcodes, or you could put the adapter sequencer straight away in your primer. So like here you have the NC1 primer followed by the barcodes, followed by on the 5' end the adapters. Like you could make the construct and have as a single primer. So in that case, like I take the DNA sample from animal 1 and use this primer with the barcodes and the adapters together and run the PCR in one go. On the other hand, I do add the adapters at the end of barcoding PCR. So either way you could do, but the concept here is like it is possible to add the adapter and the barcode for DNA extracted from each animal and run the PCR. And in that way, like finally you have a sequence where you have the ITS2 and the primary information, barcodes for each animal, and the adapters, all this added. So the next step is sequencing. And generally people use Illumina mass sequencing in that like you group all the amplicons from all animals. Like in this case, I have 3 animals. Like I group all the PCR products from all the animals and run through the Illumina sequencing. And once the Illumina sequencer reads the sequence, it also reads the barcode information, and based on the barcode, then it segregates all the amplicons. And you could see like all the green barcodes are segregated as 1, and the purple barcodes are segregated to 2, and the brown as 3. So it segregates everything and then gives the sequence information for all the animals separately. So this is how the deep amplicon sequencing works. Like you pull everything together, run it through your sequencer, and then you get your results. But in my case, I don't have a mass like Illumina mass sequencer in my lab. It is expensive to hold on to this, and I need to spend my fortune to get this mass Illumina sequencer specifically for doing parasitology work. So I need to depend on another lab, or I need to use a core facility to get this done. But to simplify that, what technology is available now is called the Manigan Nanopore Sequencing. So this is a Manigan, which is a small version of Illumina sequencer, and you could buy this and you could connect to your laptop and you could do all the things that Illumina sequencer does using this Manigan sequencer. So basically the step to get the PCR products with the barcodes and the adapter is all the same, but the adapter that we use for Manigan is different. So you need to design a primer or use the adapter specific for Manigan sequencing. And then once that is done, like you throw all the PCR product in a small deposit here, and then you deposit the sample and you run, and within 6 hours like you would get all the results. And that results again based on the barcodes. So you get all the sequence information segregated for each animal, and then you know what PCR product, amplicon goes for which animals now. So and how this is done in under Manigan. So the Manigan technology, as I said, you pull all the DNA sample and you deposit that in the to the or load them into the Manigan. And each of this sequence, like the amplicons, has the adapter sequence, which is pointed out here. And this is a nanopore sequencing technology. So on the platform, on the Manigan platform, you find a lot of these nanopores, and these nanopore then guide all the amplicons to pass through the nanopore. And once the sequence pass through the nanopore, then it reads the information present in the sequence, and these are then read through the Nemo, which is the software that kind of reads all the sequence information present in each of those amplicons, and based on the barcodes, again, they're all segregated. So this is a simple mechanism for which you could use for sequencing and get the results in one go. So how the results would appear? So the deep amplicon sequencing results, whether you're using Illumina or Manigan for the sample, like this is how the results would go. So I have used samples from farm 1, farm 2, like this, like from 6 farms. And then in each farm, I took 3 animals, and in each of those animal, like here are the list of parasites and the percentage in which like it appears. For example, Uperia, which is in blue, so it is present. Like you could see how it differs between each farm and between each animal. And then Himancus placii, like which is a cattle parasite, you could see it is only present in one of the farm and absent in others. So this is basically an example of how you would get the results of a Nemo Biome using either Illumina sequencing or a Minayan approach. So based on that, I'm just concluding my presentation on the updates or the advancement in clinical parasitology. And just wanted to thank my lab folks here. So this is my team that works in the diagnostic lab, and my mentors here, Dr. Dwight Bowman, and then Diego is my collaborator on the Minayan project. And I have whole other collaborator, including Laura Goodman, who is helping with the nanoscale PCR for tick disease identification. And all my grants in the lab are funded through USDA and NIFA, and then the National Center for Veterinary Parasitology, and then through Whole Foundation. So thank you for listening, and now I open this forum for any questions.
Thank you, Dr. Mani, for a wonderful and exhaustive presentation. I think you have taken the students from the basics of veterinary parasitology, and then telling them something about the advances that are taking place, rather the updates on the Nemo Biome, and then telling them about the importance of ticks and molecular techniques to identify the tick-borne diseases, and so on. So, students, before you go with the questions, I'll be posting a feedback link form in the chat box. So those of you who are going to see the feedback link, if you click that, it will take you to a Google form, and if you are giving a feedback about the lecture, you will automatically get a certificate within 2 to 3 minutes in your mail. So that is how it goes. And maybe if some of you want to interact, you can interact with the speaker. And for most of you do not know that Dr. Manigandan, when he started to speak to you, it was 6:45 a.m. in US. He has woke up so early, and then he is talking from there right in the early morning. So 6:45 a.m. and our timing is totally different. Almost 10 hours we are going ahead, and they are very behind. So you'll have to appreciate the fact that he has joined so early to give you his experience on the updates in veterinary parasitology. So if any of you would like to interact, you can interact. People who find it difficult to interact in English also can ask him in Tamil, because he can also reply to you. So it's over to you. Any student who want to interact, you can interact. Maybe I'll request Hari Vignesha. You'll have a lot of queries. Hari Vignesha, and then there should be Divyanandan, who is very much interested to go and do his studies abroad. Maybe some of you can interact, if not all of you. Mani, there is a question. What encouraged you to do masters in parasitology, sir?
So when I was doing my undergrad at Pondicherry Veterinary College, I was blessed with teachers who kind of instilled my interest in parasitology. For me, I need to see things to understand. And for example, no offense to bacteriologists or virologists, when they were teaching, I was struggling to understand how a bacteria, how a virus would look, how a rabies virus, they call it as a bullet-shaped virus, how it would look, all this confusion. But with parasites, when they were talking about ticks, I know they showed a tick, and then I can relate what they are teaching by looking at with my naked eye, and that's how I got interest in parasitology. So when I did my JRF, the ICR exam, I know I secured the rank, but a lot of people were asking me what subject you wanted to take. I told them parasitology. So people discouraged me about parasitology, you have rank, you should go for molecular or whatever, they were telling me, but I said okay, no, let me do parasitology. Maybe I was lucky, I'm sitting here now confidently telling that I made the right decision. What, like, if I didn't get this position, get this job, like, if I'm working as a research associate, I don't think I would advise you guys to do parasitology. But what I'm telling is like just follow your passion. Like if you know that's your interest, and I think that would guide you to success.
Okay. Anyone else would like to interact with the speaker? Instead of putting it in the chat box, you can also talk to the speaker.
Maybe they are feeling shy. Like I'll just talk a few things about what you guys can do here. Recently, the American College of Veterinary Microbiologists, when I say recently, that was in 2012, they started offering this board certification exam. So it's very similar to NET exam that you guys do, the ICR NET exam for parasitology. It's a very similar thing that they do here under the auspice of American College of Veterinary Microbiologists. So what you need to do, like you may have your DVM in India, like which is DVSC, equivalent to DVM here in North America, and then you can do a master's in parasitology, and that's enough. So once you have that, like you could take this exam. So you don't need to be here in North America, but for taking that exam, you have to travel to North America. Once you come and take that exam, it's a two-part exam. So first is general exam, second one is specialty. The general exam consists of 100 questions, I mean 240 questions, and the specialty exam is 100 questions each. So once you take the exam and once you clear it, like you would be called as a diplomat in the field, and with that diplomat status, like you could apply for jobs anywhere. So a lot of faculty jobs here in North America, they are looking for these status. Like any applicants with the diplomat status, like they would just welcome you and offer the job. And in industry, like pharmaceutical industry, like if you are a board certified parasitologist, like you would easily get a job. Like industry jobs are well paid, like Zuarez or Elanco, like a lot of those companies, Bayer, like they just take you, and not many people are in the field now. So like veterinary parasitology used to be a very strong subject, traditionally a strong subject 50 years ago, but after the discovery of ivermectin, I would say, like not just kill the parasite, but it also killed the discipline of parasitology in North America, not just North America, all across the world. So a lot of universities which used to have department of parasitology, they are all gone. But now, because of resistance emerging for most of these drugs in market, so industry is now feeling like we need to have parasitologists with classical training, or with even molecular understanding. Like they are looking for those folks. So it's the right time. Like if anyone is having interest in parasitology, the next 20-25 years is looking really good. Like if you have any desire to move to North America and wanted to pursue a degree in parasitology, especially diagnostic parasitology, and there are high chances that you would succeed in your career.
Thank you, Dr. Manigandan. Friends, there is a problem with the feedback link. I will be sending it to the WhatsApp group. I have the group of all the registered participants. I'll be sending it to your WhatsApp group, so you can fill that form to get your certificate. So thank you so much, and I would like to thank you, Mani, on behalf of the IDP scheme of TANUVAS, which is a scheme that is funded by the ICR through World Bank. And we are conducting a series of animal health lectures. And the day after tomorrow, we have Dr. Sheila from North Dakota State University. I think I told you about her. She's going to talk on new generation vaccines, truths and myths. So that's going to be for the second year students who are undergoing microbiology. And following that, we have Dad Bennett, who is going to talk about clinical pathology of small animal vector-borne diseases. That's for the pre-final students. That is going to happen on the 29th. So thank you so much once again for joining and then enlightening our students on the updates in veterinary clinical parasitology. Thank you.
Thank you so much, sir. Thank you for having me.
Yeah.